So I've combined sections 4 and 5 for the purposes of this PowerPoint. So as you're reading through your textbook, just expect that. I've thrown all the learning outcomes for both sections up here. Okay, we're going to talk about enzymes and how they're specific to certain substrates. We're going to talk about how those enzymes bind to their substrates, what can affect how well enzymes work, and then we're going to talk about different kinds of metabolic pathways and the reactions that we find in metabolism. Okay. Now, with a exception that we will get to later in the semester, enzymes are proteins, right? Except for the one weirdos that we're going to talk about later. We know that protein structure is really important to its job, right? When we twisted up those little pipe cleaners, we talked a lot about how form follows function. If we need a protein to do a job, it has to be a specific shape, okay? And this is very true for enzymes, right? So if we remember the quaternary or the tertiary and the quaternary structure of proteins, where we have our three-dimensional shape and then we have all these different subunits coming together to make up the quaternary structure, that those levels of organization determine the job that those enzymes can do. Okay, and we need, in order for an enzyme to function, the active site. So here's our little gray circle blob thingy. This is representing our enzyme. What we're going to see is the reagents that are the reactants that are coming in to have our chemical process go. We call that thing the substrate, and that substrate has to fit into this special site called the active site, right? So we have on our enzyme we have a pocket, and so our active site pocket has to be the right size and shape for our substrate to fit into. Right, so if I can't fit, I'm going to have a really hard time fitting this fire extinguisher into my pocket because my pockets aren't meant to hold fire extinguishers. Why would they? That's weird. Okay. So the active site of an enzyme has undergone this evolutionary process to fit the substrate, the size and shape of whatever the starting point is for that chemical reaction. Okay. And if there's something that occurs, whether that's a mutation in the DNA, right, the instruction manual for building a protein, or if there's a change in the environment that changes the tertiary, the quaternary structure of that enzyme, that active site might be different and it won't fit that substrate anymore. So let's look at this real life example. So again, when we get back to class on Monday, we're going to do a lab where we are going to intentionally mess up our boy lactase. Lactase is an enzyme that is going to take our lactose sugar, and it is going to break it from a disaccharide into two monosaccharides, specifically glucose and galactose. Okay, so the active site of lactase has to fit the three-dimensional shape of lactose. Okay, so now you guys can tell me what the substrate for lactase is and what the products for lactase are, because I just told you. I'm not going to tell you again. Now I sound like your mom. All right, so again, the way that enzymes are going to lower that activation energy is that they are going to tweak the structures so that it's easier for those bonds to break, or right in the case of a catabolic enzyme, which is what I'm showing here, or we can also have enzymes that do the anabolic process that kind of line everything up so it's easier for those bonds to form. So let's look at this one. So here we have our enzyme sucrase. Okay, sucrose is the substrate, and sucrose is a glucose and a fructose molecule bonded together. What this enzyme is going to do, oh, I certainly don't have enough hands. It's the sucrose is going to land into that active site. That interaction between the amino acids in the enzyme and the sugar molecule is going to change the three-dimensional shape of the protein temporarily. It's going to stress those chemical bonds and make it easier for those bonds to break. And that means that there's going to be less activation energy required to break that bond. Because if they're hanging out here, like they're happy, they're satisfied. But you put a little bit of pressure on it, and it starts to fall apart. Hey, don't call me out, me. Okay, so let's look at this. This is really important that the enzyme that we end up with is the same in the chemical structure, right? The three-dimensional structure that it was when we started. Okay, so our enzymes are not going to get consumed or changed permanently during the course of this reaction, right? We see a temporary three-dimensional shift, but as soon as those that bond is broken in this case, or formed in the case of an anabolic enzyme, right, then it's going to go back to its shape it was before, so that it can now just be ready for the next thing. So I kind of think of enzymes as checkers at the grocery store, right? So if you've ever worked as a checker at the grocery store, like I have, right, you just see a lot of people and you're just like beep, 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 right? And you're handling all their stuff and you're maybe like doing some small talk with them, and you have this kind of temporary interaction. As soon as they're gone, your check stand structure is still there. Everything's ready to go for the next person. Okay, so this is kind of just how enzymes are. I forgot to say this, so I'll say it now that I remember. So when our substrate binds to that active site, we get this enzyme substrate complex. And again, that's that temporary stressing of those bonds. In this case, in a case of a catabolic reaction, our enzyme is going to alter its three-dimensional shape slightly, have that reaction occur, and then go back to its normal. All right. What we tend to see, though, is not just one enzyme hanging out by itself. Usually, especially for our really important processes that are essential for life, we usually see a lot of chemical reactions that are required. And so we see enzymes in this big long chain called these multi-enzyme complexes. Okay, and so we're going to actually have like one group of stuff that is actually several enzymes that are going to catalyze different reactions in this stepwise process. Okay, so let's have a look here. All right, so the enzyme, our first enzyme is going to use the substrate of yellow triangle, and its product is going to be green square. Enzyme 2's substrate is going to be, oh, that's a rectangle. Let me say that again. Okay, so enzyme 1's substrate is yellow triangle. The substrate for enzyme 2 is green rectangle. The product for enzyme 2 is blue trapezoid. The product for enzyme 3 is orange square. Okay, so we see these intermediate substrates, and those intermediate substrates, they're the product of the enzyme before. Okay, so we're like, we're assembly line in this, right? So the first thing that you want to do, let's see, what's a good example that I can pull off the top of my head? I don't know. Let's do packaging beer, because that's something that my husband does. So you have to like, so when they can beer, there's 3 or 4 guys. One guy is to make sure that all the cans end up on the right pallet, and make sure that they're loaded in the machine properly. Okay, and then the next guy makes sure that the cans are getting washed and sterilized properly. The next guy's job is to make sure that the cans are getting filled properly. The next guy is going to be making sure that the lids are put on properly. And then the next guy is going to make sure those lids are sealed. And then the next guy is going to put the little plastic 6-pack holders on the beers and stack them up on the pallet. Okay, we'll just use that as an example. Okay, so this line is all happening in a series of reactions. You can't put the lid on the beer before you put the beer in the can, right? That would be silly. And you want to have all of these different processes be like close together spatially so that it's really effective, right? So you're not like running from this side of the brewery to that side of the brewery for that step, and then running over here and then running over there, right? You're just going to like line these things all up so that you can be really fast and effective with this process. Same thing is happening on the cellular level, right? On this protein level, where we're taking our substrate, we're doing our reaction, we're taking that product, we're handing it to the next person in line. They're taking it, doing what they need to do, handing it to the next person in line, and so forth. Okay, and we're going to play a game like this, so if it's not clicking, it will later, I hope. Okay, so the benefits of having these multi-enzyme complexes, right? So again, keeping everything compact in space is going to prevent those products from diffusing away. It's going to make sure that we are not having these unwanted reactions, like we don't have other stuff sneaking in and getting in the way, so that we can increase that efficiency. And it's going to also allow us to control this complex as an entire unit rather than all of these individual interactions. Okay, so real life example of this, and again, we'll see this enzyme when we look at cell respiration. Here we have pyruvate dehydroxygenase. That doesn't make any sense to you right now, and that's okay. We're not freaking out about that. We'll see it later, and it'll kind of click free then. But what we need to know is that there's actually 60 polypeptides that are creating this enzyme complex. And this one enzyme complex is actually catalyzing 7 different reactions. So the first one's coming in, it's changing it, passing it along in this line, even though this looks like one unit. So we can see what this looks like at the model level, right, where we have all these different active sites. And then this is what it looks like using a transmission electron micrograph, a TEM. Okay, so this is what it actually looks like in a cell. All right, so when we're talking about enzymes, it's really important to remember that form follows function. And we are going to, in our lab, we are going to intentionally mess up our enzyme lactase using a variety of environmental factors. Okay, so some factors that can affect how well enzymes do their job are temperature, pH, the presence of these things called inhibitors and activators, and also coenzymes or cofactors. Okay, so let's go through each one of these individually. All right, so each enzyme is going to be adapted to a specific range of temperatures where it's going to be the most efficient. Okay, so if we look at this, trypsin enzyme, its job is to break proteins down. For humans, the most effective temperature where we get the highest number of reactions is about 40 degrees Celsius. Our body temperatures for most humans are 37 degrees Celsius. So we can see that, yes, our optimal temperature of this particular enzyme is the same as our body temperature. We take that same enzyme and we look at it in an organism that lives in the hot springs from out over in Yellowstone. And we see that its optimal temperature is somewhere around, I don't know, between 72 and 74 degrees Celsius in those prokaryotes because they're adapted to much hotter environments. If you take our enzyme from our humans and we put it in that 70 degrees Celsius environment, that protein is going to denature, right? Here's our, again, let's use this dog toy for another analogy, right? It's supposed to be this shape. It's going to get smooshed. It's going to change shape. It's not going to do its job, and it's going to get messed up. Okay. Similarly, if you took the enzyme from that archaea and put it into human body temperature range, it's going to denature. It's too cold for that protein to do its job. Okay, so these temperature extremes that are beyond its normal can really affect how well the enzyme performs. Okay, we're going to do that with lactase, and we're going to see if we can denature lactase with using temperature. So pH also, so the interactions between when we have a strong acidic or a strong basic environment is going to determine the three-dimensional structure of the enzyme. So here we're looking at our trypsin, and another enzyme that's used. So pepsin is an enzyme that's used in digestions. Pepsin is found in our stomachs, and trypsin is found in the small intestines and the pancreas. So even though they're both involved in digestion, we find them in different places. And as you probably know, right, your stomach has really low acidic environment. Its pH is typically between 2 and 3, which is the optimal pH level for pepsin. If we took pepsin and we put it in our pancreas or our small intestines, it couldn't do the job. It couldn't catalyze that reaction because it would be denatured. Okay. Same thing with trypsin. It needs about a neutral-ish pH. If we put it too acidic or too basic, it's going to denature. Okay. So this is another concept that we're going to draw on the whiteboards when we get back into class. Okay, so we're going to have our competitive inhibition. So our enzymes, again, right, have that active site. That active site is the pocket that's going to fit our substrate. If we have a competitive inhibitor, this is going to be a non-target substrate, right? So our inhibitor is not our substrate, it's something else. But what it's doing is it's sneaking in and getting into that active site before the substrate can get there. And so if this inhibitor is present, the substrate no longer can fit into that active site, and we have a lower, less enzyme activity. Okay, where we see this and we're going to see this again when we get to our photosynthesis chapter. We're going to look at this enzyme called rubisco that is going to take the carbon dioxide from the air and catalyze that reaction and bring it in, and we're actually going to stick it onto a 5-carbon sugar. So the carbon dioxide is the target substrate, but oxygen can also fit into that active site and block carbon dioxide coming in. So we'll see that again later in the semester. Okay. So in a non-competitive inhibition, our inhibitor is not binding to the active site. It's binding to the allosteric site, and the allosteric site is the other site, right? It's the other place. It's right. So if our active site is our front pocket, the allosteric site is the back pocket. Okay. So this inhibitor is coming in to the allosteric site, docking onto that back pocket, changing the three-dimensional shape of the active site, and now the shape of the active site no longer fits the shape of the substrate, and we don't have that chemical reaction being catalyzed anymore. Okay, so make sure that you know the difference between competitive inhibition and non-competitive inhibition, because these are things that I like to have you guys draw on exams. Ow, that hint just landed on my foot. All right, so another thing that can kind of trip students up is this idea of allosteric activation. So let's go back. So in allosteric inhibition, right, we have our inhibitor landing in the back pocket of the enzyme. It changes the three-dimensional shape of the active site, turning off the enzyme, right? So that substrate no longer fits into that active site. That enzyme is not doing its job, and we're not catalyzing that reaction, right? So inhibition is the off switch. In allosteric activation, when the enzyme is just hanging out by itself, the active site shape does not fit the substrate. And we need that activator, right, that to come in and to land in that allosteric site to change the active site so that the substrate does fit. So allosteric activation is like turning that enzyme on. Okay, so make sure you know the difference between allosteric inhibition and allosteric activation. Okay, so if we go back to this idea of these multi-enzyme complexes, right, where we have this long chain of all these intermediate enzymes getting these intermediate substrates, what typically happens is that our final product, right, whatever the thing is that we're trying to make, the whole reason that we have this big long complex usually will be a allosteric inhibitor of that original enzyme. So when we have a high enough concentration of the product, right, when we've canned enough beer, we can shut down the beer canning process. We don't need to make more because that will be wasteful. We're going to waste all the energy that's required for all these reactions to occur, right? And if this product has a short shelf life, then we're not like being efficient. Okay, so having the product shut down that original enzyme is actually a really good strategy for making sure that you're conserving energy and conserving resources. All right, so the other thing that we just need to talk about real quick are cofactors and coenzymes. There is like some actually hair splitting differences between cofactors and coenzymes, but that's to a level that we don't need to really get into this class. But what I do want you guys to know is that cofactors are non-proteins, so these can be organic compounds or inorganic compounds that are needed for the enzyme to function. Okay, and they're not like, not every enzyme needs a coenzyme or a cofactor, but some of them do. So an example that we'll talk about is the when we're taking our glucose molecule and we're breaking it down so that we can produce ATP. In one of the steps, we need a coenzyme that's going to take that that partially broken down sugar molecule and move it from the cytoplasm into the mitochondria. And to do that, we need to have a coenzyme, because otherwise, without this other little bit here, our active site doesn't actually fit the substrate. So we need the enzyme and the coenzyme together, right, to form an active site that the substrate will fit. Okay, IRL example we'll see during cell respiration. All right, so some key takeaways for this chapter, right? So the sun is going to provide the energy for living systems. ATP is going to be the energy currency of the cell. So all of our cell processes are going to be powered by ATP. First law of thermodynamics is energy is not created or destroyed, it just changes forms. And the second law of thermodynamics, entropy is always increasing. So shit's messy and it's going to keep getting messy. Where you know that all chemical reactions require activation energy, but we can also have catabolic, which are endergonic. We can have catabolic reactions that are exergonic, where we're releasing energy. we can lower that activation energy by using enzymes, and enzymes are specific to their substrates because form follows function. Okay, make sure and check out these visual summaries. They're mighty fine study aids. And let me know if you guys have any questions. I'll see you on Monday.